All right, everybody. Welcome to another deep dive.
Yeah.
I love getting into the weeds on these topics.
Oh, yeah.
And today we're talking all about secondary antibodies and how important the incubation time is.
Absolutely. And it's interesting because, it's such a simple concept,
Yeah.
but it really underpins so much of biological research. And it doesn't matter if you're doing Western blots, ELISA's immunohistochemistry,
Right.
getting that incubation time right is critical for the assay.
For sure. For sure. So we've got a great article today from Assay Genie that's going to kind of guide our discussion.
Yeah, that's a good one.
And we're going to pull out some of the little details that they highlight.
Yep.
But before we get ahead of ourselves, let's take a step back.
Okay.
For folks who might be new to this, can we just kind of lay the foundation?
Sure.
What are secondary antibodies? Why are they important in these kinds of immunodetection techniques?
Okay. So imagine you're trying to track down a specific protein in a sample, right?
Mm-hmm.
Your primary antibody is your first line of attack.
Okay.
It binds directly to that protein. Right.
But a lot of times the primary antibody itself isn't detectable.
Gotcha.
So that's where the secondary antibody comes in.
Okay.
And it acts like a beacon. It attaches to the primary antibody and carries a signal that we can detect.
Okay.
So that might be an enzyme or a fluorescent dye or something like that.
So it's like the primary antibody points and the secondary is like, hey,
exactly.
Here it is.
Yeah, it's like, hey, over here.
Yeah.
So we can see it.
I like it.
And that whole process is called signal amplification.
Okay.
Because one primary antibody can actually trigger a cascade of secondary antibodies,
Interesting.
making the signal much, much stronger.
So the article called it a chain reaction, which I thought was a good way to think about it.
Yeah.
Like a molecular game of telephone.
Exactly.
Where the signal just keeps getting louder.
Yeah. And the time part of this comes in during the incubation step,
Right?
which is the time we allow
Okay.
for those secondary antibodies to find and bind to the primary antibody. Gotcha.
If it's too short, we might miss some of the signal.
Okay.
But if it's too long, we risk those antibodies binding to things they're not supposed to.
Okay, so it's like a Goldilocks situation.
Exactly.
Not too short, not too long.
Yeah.
But how do researchers figure out the right amount of time?
Well, it's a great question, and it's definitely not one size fits all.
Okay.
There's a bunch of factors that influence the ideal incubation time.
Okay.
And one of them, for example, is the concentration of your secondary antibody solution.
Okay, so if you think about this like a matchmaking game.
Yeah.
Higher concentration, more potential partners bumping into each other.
That's right.
So does that mean then that researchers always want to go for like the highest concentration they can?
Not necessarily.
Yeah.
While a higher concentration can definitely speed up the binding process, it can also be like a crowded dance floor.
Okay.
If it gets too crowded, those antibodies might start getting a little too friendly with the wrong partners
Right.
and bind to things they're not supposed to.
Yeah.
And then we start to get that noise in the background.
Gotcha. So it's a balancing act
Exactly.
between speed and accuracy.
Yeah.
What other things are researchers thinking about?
Temperature?
Temperature.
Is another big one.
Right.
So think about if you heat up a pot of water,
Yeah.
the molecules move around faster. They bump into each other more frequently.
Right.
Same thing with antibodies.
Okay.
Warmer temperatures generally speed up the binding process,
Okay.
which allows for shorter incubation times.
And I'm guessing cooler temperatures would do the opposite.
Exactly. It's like turning down the thermostat
Okay.
on all those molecular interactions.
Gotcha. This is all making sense.
Yeah.
But I'm wondering about one thing the article mentioned, the type of assay
Yes.
can influence the incubation time.
Right.
Can you explain why that is?
Sure. So think about it like you're trying to find a document
Okay.
in a library.
Mm-hmm.
So in some assays, like a Western blot,
Okay.
the target proteins are nicely organized on a membrane. It's easy to access.
Right.
But in others, like immunohistochemistry, those proteins are hidden away in the tissue section.
Okay.
So it's like the difference between finding a book on a shelf
Right.
and digging through a whole archive.
Okay.
The harder it is for those antibodies to reach their target, the more time they'll need.
Okay.
So that's why Western blots typically have shorter incubation times
Gotcha.
than something like immunohistochemistry.
Okay, that makes sense.
Yeah.
So we've got concentrations, we've got temperature, we've got the type of assay.
Yep.
What about the target itself?
Yeah,
Does the amount of protein that you're trying to detect matter at all?
it absolutely does.
Okay.
So if your target protein is abundant in the sample, it's like a bestseller
Okay.
on display in a library.
Gotcha.
Those antibodies will find it quickly.
Right.
But if it's a rare protein,
Yeah.
then you're going to need to give them a lot more time to hunt it down.
So it's not just following a recipe.
Bro.
You need to understand all these factors to tailor your approach.
It's all about context.
But how do researchers actually find that perfect incubation time for their specific experiment?
Well, there's definitely some trial and error involved,
So-
but it's more systematic than just guessing.
Okay.
The article calls it empirical testing, which is essentially like fine-tuning a recipe.
Yeah, sure.
You start with a basic framework and then you make adjustments
Okay.
based on your observations.
All right. So let's say I'm a researcher. I'm doing a Western blot.
Okay.
I've got my antibody concentration. I got my temperature. I know the abundance.
Yeah.
Where do I begin?
Well, first, you want to consult any existing literature or protocols.
Okay.
There might already be some recommendations for incubation times,
Okay.
but from there, it's all about experimentation.
Okay.
You know, you might start by testing a range of times, maybe 30 minutes an hour, 2 hours.
Okay.
Compare the results.
So you run the experiment with those different times, and then you look at the data
That's right.
and see which one had the clearest signal
Exactly.
with the least noise.
Yeah, and there are actually a few tricks of the trade
Okay.
that can help you optimize this process even further.
Okay.
The article mentions something called blocking agents.
Blocking agent.
Have you heard of those?
I mean, just based on the name. I'm guessing they block something?
Yeah,
Do they, like, prevent the antibodies from binding where they're not supposed to?
you got it.
Okay.
Blocking agents help reduce that unwanted noise
Okay.
by occupying those non-specific binding sites.
Lucha.
So that gives the secondary antibodies a clear path to their target.
So it's like those traffic cones
Yes.
that they put up on roads
I love that analogy.
to direct the cars in the right direction.
Yeah, it highlights how much thought and strategy goes into this.
Totally.
These seemingly simple techniques.
And what I thought was really cool about the article is they actually cited research studies
Yes.
to back this up.
Yeah,
It's not just theoretical.
absolutely.
There's data
Real data.
to support all of this.
Yeah.
Cool. So we've got the basics down,
All right.
but there's so much more to this than meets the eye.
Yeah, definitely. Welcome back to our deep dive on secondary antibodies. And, you know, we were just talking about how researchers go about figuring out that sweet spot for their incubation times.
Yeah, like, it seems like there's a lot of pieces to this puzzle.
There are.
Antibody concentration, temperature, the type of assay, the abundance of the protein.
Exactly.
How do you actually bring all of that together to figure out, okay, this is the right amount of time?
Well, it really comes down to understanding the dynamics of how those antibodies and antigens interact with each other.
Okay.
And the article dives into this concept of kinetics,
Kinetics.
which I know sounds intimidating,
Yeah.
but it's actually pretty straightforward once you break it down.
Break it down for me.
So essentially, kinetics is the study of how fast those reactions happen
Okay.
and what factors can speed them up or slow them down. Think of it like you've got these secondary antibodies floating around, right? Their mission is to find and bind to the primaries.
Okay.
Kinetics helps us to understand how efficiently that happens.
So it's not just if they bind,
No.
it's how quickly they do it.
Exactly. And one of the big players in all of this is something called affinity,
Affinity.
which is basically a measure of how strongly the secondary antibody is attracted to that primary antibody.
Okay, so high affinity means they're basically like meant to be.
Soulmates.
Yeah.
A high affinity antibody will bind quickly and tightly, which means you could probably get away with a shorter incubation time
Gotcha.
because those antibodies are going to find each other really efficiently.
That makes sense.
Oh,
So what if you have like a lower affinity antibody?
right.
Do you just need to increase the incubation time then to compensate?
You could definitely try that.
Okay.
It's like giving those antibodies more time to mingle and find their match.
Okay.
But sometimes even with a longer time, that lower affinity antibody might not bind as strongly,
Okay.
and that could lead to a weaker signal, which might be hard to detect.
So choosing the right antibody is like choosing the right dance partner.
Exactly.
You want someone who can keep up with you
Absolutely.
on the dance floor.
I love that. And just like on that dance floor, the concentration of antibodies also affects how quickly they find each other.
Right.
So a higher concentration, again, more chances for bumping into their targets.
Right. But too high of a concentration and you've got that risk of like
Exactly.
too much noise.
Too much noise.
So how do you balance affinity concentration and incubation time?
Right. Well, that's where it gets really interesting because the optimal time isn't just a fixed number.
Right.
It's a product of all those things together.
Okay.
The affinity of the antibody, the concentration you're using, the temperature,
Right.
and even the characteristics of the protein you're trying to detect.
It's a lot of variables
It is.
to take into account.
Yeah, the article uses this phrase, time-dependent signal amplification,
Time-dependent signal amplification.
which basically means that the longer you incubate,
Okay.
the stronger the signal could become, but only up to a point.
Okay, so what's happening at like the molecular level
Yeah.
as that time is going on?
So remember that each primary antibody can have multiple binding sites
Right.
for those secondary antibodies.
Okay.
So as time passes,
Yeah.
more and more can latch on
Okay.
to the primary antibody, which increases the overall signal.
So it's kind of like you're building a tower.
Yes.
The more time goes on, the higher the tower gets.
Perfect analogy.
Okay.
But at some point you reach a limit, right?
Sure.
Where all the binding sites are occupied,
Okay.
and adding more time isn't going to make the signal any stronger.
It's like trying to add blocks to a tower
Exactly.
that's already full.
Yeah, and if you go too long,
Yeah.
you can actually start to get the opposite effect, where those antibodies might start binding to things they're not supposed to,
Right.
which increases that background noise.
So it's like this balancing act between maximizing the signal but minimizing the noise.
Exact.
And the incubation time is how you achieve that.
That's right, and that's why the article emphasizes this empirical testing.
Right. We touched on that before.
Yeah.
Can you remind us what that means?
Yeah, so empirical testing basically means figuring things out through experimentation.
Okay.
It's like the scientific method of trial and error.
Gotcha.
So you start with a hypothesis,
Mm-hmm.
maybe based on some guidelines,
Okay.
and then you just test it out.
Okay.
So you would try different incubation times and see what gives you the best results.
So if I'm a researcher, I try 30 minutes an hour, 2 hours,
Exactly.
and then I see which one gives me the best result.
That's right.
And that's where having that deep understanding of like the affinity, the concentration, the temperature,
Yes.
all comes into play.
Exactly. It helps you interpret the results and make decisions about how to adjust things.
It's like detective work.
It is.
You're gathering all these clues
Absolutely.
to solve the case.
And speaking of clues, remember we talked about how the type of assay
yeah.
you're using can also have an impact?
Yeah, like Western blots have shorter times
Right.
than immunohistochemistry
Exactly.
because the proteins are more accessible.
Yeah, it comes down to how those proteins are presented.
Okay.
So in a Western blot, they're laid out nice and neatly on that membrane.
Okay.
But in immunohistochemistry,
Yeah.
they're embedded within the tissue.
Right.
So it's much more complex
Yeah.
for those antibodies to navigate.
It's like trying to find a file on your desktop
Yeah,
versus like digging through a whole hierarchy of folders.
exactly. In immunohistochemistry, those antibodies might have to weave their way through cells and all kinds of things just
Yeah.
to reach their target.
So they need more time.
They do.
Right.
And the article emphasizes that these are just a couple examples. It really depends on your experiment.
It's like each experiment is its own little puzzle
It is.
that you have to figure out.
And that brings us back to the power of that empirical testing.
Okay.
It allows you to fine-tune everything
Gotcha.
based on what you're actually doing.
Cool. Well, this is feeling a lot less intimidating now.
Ugh.
It's more like we're learning this secret language of
Yes,
antibodies.
That's a great way to put it. And speaking of exciting challenges, remember that question we had
yeah.
about climate change?
How could climate change possibly affect
Right.
antibody incubation times?
Well, let's think about it.
Okay.
Remember, temperature plays a role.
Yes?
So if labs are experiencing more extreme temperatures because of climate change,
Yeah.
it could be really challenging to get those consistent results.
Okay, so let's say a lab is in the middle of a heat wave.
Right.
They're trying to do this experiment. What kind of problems might they run into?
Yeah, so we were talking about labs in a heat wave.
Right. What's going to happen to their experiments?
Well, warmer temperatures, they tend to speed up those reactions.
Mm-hmm.
So if a lab isn't super carefully controlling that temperature,
Yeah.
things could happen a lot faster than they're expecting.
Okay.
You could get overbinding,
Okay.
where the antibodies start glomming onto things that they shouldn't.
Right, so they get a little overexcited.
Exactly. It's like they're getting too friendly on that crowded dance floor.
Okay, so more noise in the data.
Exactly. And then you've got the opposite problem.
Yeah.
In places that are getting colder.
Right.
Things might slow down too much.
Yeah.
So then they need to increase that incubation time
Okay.
to make up for it.
But then how do you compare
Right.
experiments that are done
Exactly.
in different parts of the world?
That's a really good point.
If the temperatures are different,
Yeah.
are the results even comparable?
Well, and this is why it's so important to document everything really carefully.
Okay.
Researchers need to be upfront about those conditions, especially the temperature,
Yeah.
so that other people can interpret the data correctly.
So it seems like labs are really going to need to up their game
Yeah.
when it comes to temperature control.
I think so. I think we're going to see even more of those super precise incubators.
Yeah.
You know, the ones with all the bells and whistles.
Oh, they.
Because controlling that temperature is going to be even more important.
And maybe even like new technologies.
Yes.
New antibodies that can withstand a wider range.
That's a great point.
Of temperatures.
I think there's going to be a lot of innovation
Yeah.
to overcome these challenges.
It's pretty amazing.
It is.
To think that climate change could impact something like this.
It's all connected.
It is.
And I think that's what makes science so fascinating.
Yeah. Well, this has been a really eye-opening deep dive.
It has.
Into the world of secondary antibodies.
A lot more to it.
Than I think we realize.
Yeah, than meets the eye.
At first glance. So big thanks to our expert for walking us through all of this.
Oh, it's been my pleasure.
And to everyone listening.
Yeah.
Be sure to check out that article from Assay Genie.
It's a good one.
For all the details.
Yeah.
And as always, stay curious.